Introductory Neuroscience and Neuroinstrumentation, ERPs, but different event-related potentials. Hello. In this session, we shall introduce different event-related potentials. Knowing about different ERPs and their components can help determine questions that can be tackled with appropriate ERPs. A psychological or neural process can be studied if variations in that process lead to a measurable change in the ERP. Okay. So this is just a brief overview of commonly used event-related potentials. There are many more, but we can't possibly cover every single one of them. So I shall be focusing on the important ones and the ones generally used in clinical research. So ERPs, all of them are not present all the time. Some of them are present at birth, but most of them develop as the brain develops through age. So at birth, you have MMN. MMN is the first cognitive ERP present at birth. We have discussed MMN in previous lectures. You also have ABR. It's an evoked potential. It's not an event-related potential, strictly speaking, and it checks the auditory pathway integrity. MMN shows indicates acoustic regularity or short-term auditory echoic memory if it's an auditory MMN. So P50 gating is also present at birth, and this relates to attention and reduced responses to redundant stimuli. Around 4 or 5 years of age, you get the P300, where you see, any novelty in the visual environment evokes a response between 300 to about 450, 500 milliseconds. Around 10 years of age, you get the recognition potential, where you start recognizing alphabets. Around 12 years of age, you have the N400 evoked event-related potential. This occurs when there is some incongruity in verbal or math stimuli. Later, at around about 15 years, you have the contingent negative variation, which is used to evaluate how much risk-taking a subject would indulge in. Okay? So putting all these things together, you could get a brain function index, which is what I'm working on in my lab at St. John's. So definitions. So just to reiterate, event-related potential waveforms. These are changes in scalp recorded voltage over time. You record over the scalp, this is a heat map, and over time that reflect sensory, cognitive, or motor processes elicited by a stimulus. A peak is exactly what it says. You have different peaks, and the logic here, it's positive up. So the first peak is called P1, the second one P2, and the third one P3, which relates to the P300. And similarly, you have N1, N2, and so on and so forth. Now each peak may be made up of many, probably is made up of many components, and all these components merge and give rise to the peak. Okay. So again, to refresh your mind, how do ERPs occur? So postsynaptic potentials caused by neurotransmitter binding to postsynaptic receptors opens channels and causes postsynaptic potential. So if you see over here, you have the resting membrane potential, and then you have an excitatory postsynaptic potential, another one here, and then they come simultaneously and they reach threshold, and you have an action potential. So these guys, cause the EEG. This is not, is not recorded by scalp electrodes. So looking at it in a little more detail, here you have a classic pyramidal neuron without the dendritic tree, because that would make it very busy. So you have synapse over here, an excitatory synapse. You have an inhibitory synapse over here, and together these things merge and give rise to the postsynaptic activity. So consider this trace. This is the microelectrode has been put in here, and the presynaptic excitatory terminal, and each time it fires, you have an excitatory presynaptic activity. Here you have a spike here, and you have two more potentials over here. Here it's inhibition, so it's the opposite. Together, they give rise to the EPSP and IPSP. So the EPSP looks like this, the IPSP looks like this. This is recorded from the pyramidal cell. When you record from the presynaptic, whether it's excitatory or inhibitory, it kind of looks the same. Right? So if two of them merge or come close to each other, the postsynaptic cell reaches threshold and then it fires the action potential. Again, the action potentials are not really recorded on the surface, it's only the EPSPs and the IPSPs, excitatory postsynaptic potentials and inhibitory postsynaptic potentials. Where do the components of the ERP originate? For a neural generator source, the distribution of positive and negative voltages recorded on the scalp is determined by the position of the dipole in the head and its orientation. So when groups of neurons fire and they are all aligned in one particular axis, they can be considered, or they act like a dipole, like a magnet with the north and south. The positive or negative polarity of an ERP component at a given electrode site is related to several factors, including the orientation of the equivalent current dipole with respect to the electrode. So all the individual dipoles sum up and we can consider them as an equivalent current dipole. So it is important to note that the polarity of a process, whether it's going up or down, P1, N1, P2, N2, does not relate to inhibition or excitation. It may, but it may not. On the right over here, these are recordings from my lab using Neuroscan CAP. I'm just showing the midline electrodes, FPZ on the forehead, FZ, FCZ, CZ, CPZ, PZ, POZ and OZ. Okay. So as you can see, there is an inversion, and the inversion occurs if you look at this first component over here, it gets less over here, it gets even less at CZ, you can barely see it, CPZ you can't see it, PZ it comes but it's inverted, and as you go down to the occipital region it gets more and more inverted. So likewise with the second component. So here you see it going up, upward deflection, and when you go to the posterior electrodes it goes down. So we can think of a dipole, happening in this central area, which changes polarity from front to back. Okay. So ERPs, so they're direct, instantaneous, millisecond resolution of activity related to neurotransmission. Because the electrical potential travels close to the speed of light, the transmission through the brain, the meninges, the skull and the scalp is essentially instantaneous. And the voltages measured on the scalp at a particular time reflect synaptic activity at that particular instant. So if you see on the right, this is a canonical ERP representation, negative up. So N1, that's around about 100 milliseconds. It generally reflects pre-attentive perceptual processing in the primary cortex. P2, pre-attentive processing the primary cortex, and you can see if you look at the heat map where the activity shows. N1 is there, P1 is here, and N2 is also over there, and it reflects stimulus, decision, etc. N4 is semantic, conceptual processing, and here the activity moves behind to the occipital areas. P3 is the P300, it's there behind, and P600 is syntactic processing. Again, the principal thing is behind. Okay, so these are kind of averages, the consolidated sum of all the processes happening in the brain at that particular time. So one of the methods used to display data is grand averages. Here you take the averages of many subjects, so that if you look at the last trace over here, it looks nice and smooth. It's really pretty. But there is a problem over here. This is the grand average of 8 subjects. This is from a study by Stephen Luck. And you see the variation between subject 1 and subject 2, subject 3, subject 4, subject 5. It generally follows this pattern, but there are very significant individual variations. So you have to be careful. If you do grand averages, you'll get lovely statistics, but you lose all the individual variations. So if you use the individual variations, then it's very noisy data. So you have to kind of compromise, and the kind of analysis you do depends on the questions you're asking. So small differences. So consider an electrode over here on the scalp, okay, and another in another subject, electrode at the same place. So these two subjects, there's a sulcus over here. So there's slight variations. Here the dipoles are like this, so most of them on the top and a few of them below. It's opposite over here. So the resultant or the equivalent dipole over here points up, okay. Here it points down. So that's why you have significant differences between subjects, because the brain, the sulcal gyral patterns are very variable between subjects. Okay, so now let's look at the auditory brainstem response in detail. So under appropriate conditions, it is possible to observe a sequence of ERP peaks within the first 10 milliseconds of an auditory stimulus. So this is just 10 milliseconds, and you have these following peaks: 1, 2, 3, 4, 5. 6 and 7 are a little difficult to record, but this is the auditory brainstem response. And this is the basis of checking for hearing loss or auditory dysfunction in neonates. Okay. So various lines of evidence indicate that these peaks arise from the various stages along the auditory pathway. So you have a stimulus coming in here. First you have the cochlea, then you have the cochlear nucleus, then you have something called the superior olive, then you have the inferior colliculus, and then you have the auditory thalamus, the medial geniculate body, and then finally the auditory cortex. So the response from the tympanic membrane, the eardrum, to the brainstem, all these different way stations, gives rise to these potentials. So if there is a problem, one of these components would be delayed or even absent. And this is the auditory brainstem response. And again, it's between 0 and 10 milliseconds, okay, the green traces. So getting into a little more detail, where are the electrodes placed? So below is a baby. You have usually 3 electrodes, one on the forehead, 2 on the behind the ears, the mastoid prominences or the mastoid bone, and then you have a ground and a reference. And the stimuli are very different. Since it's 10 milliseconds, the stimuli are much shorter. It's 0.1 millisecond or 1 millisecond. And they're clicks. And they occur, we give them at 50 to 100, 150 hertz. So it comes like that. But the brain can distinguish, the cochlea and the brain stem can distinguish individual stimuli at that frequency. And the amplitude is pretty high, 90 dB. This, in normal hearing, it can cause damage to the ears, but since the stimuli are so short, 0.1 milliseconds, it doesn't cause any damage. And the clinical relevance is babies. Many babies are born with auditory dysfunction, and for example, they have thyroid hormone dysfunction. And if we find out that there is abnormal ABR, and then we do the bloods of the baby and find out there is a problem with TSH, T3, T4, the thyroid hormone, then we can immediately add thyroid supplements to the nutrition of the baby. And this is as simple as that. If you don't do it and this continues for months, then the baby has congenital deafness. And once it's deaf, then it's very difficult to treat it once it sets in. So the sooner we catch it, then we treat it, and the baby is perfectly normal and has a perfectly normal life. So that's why it's important. The second thing is we don't need any responses from the baby. It can sleep, doesn't matter. These responses occur even if the baby is asleep. And also sometimes it's used in monitoring auditory function during surgery if you have a brainstem tumor. So you constantly monitor the ABR during surgery to make sure that no damage occurs to the brainstem pathways due to surgery. So the ABR is followed by mid-latency potentials, which probably arise from the auditory thalamus, the medial geniculate body, and just before the primary auditory cortex. So over here, just before P1 are the mid-latency potentials. These are mainly of research interest. I haven't seen too many applications of this in clinical neurophysiology. P1 is the largest potential after the mid-latency potentials. It occurs around about 50 to 60, 70, 80 milliseconds, and it arises from the primary auditory cortex, and it's largest at fronto-central sites. Okay. So this is a classic auditory evoked potential, and you have the P1, and then you have the N1 and P2. Okay. So coming to the N1. So N1 occurs around 100 milliseconds, and this peak has several subcomponents. So one component is a frontocentral component, that's over here, and it peaks at around about 75 milliseconds, and it's generated in the auditory cortex. Over here, this is approximately the auditory cortex. The primary auditory cortex is inside this Heschel's, this Sylvian fissure over here, and the stuff on the outside is Wernicke's area and Broca's area and stuff. So these are all parts of the auditory system. And following the frontocentral component at 75 milliseconds, you have a vertex maximum potential that occurs around 100, and we don't know where this comes from. And then you have a laterally distributed component which peaks at around about 150, which is generated in the superior temporal gyrus. That's over here inside. Okay. So all these components, these three components, merge, and together they give the N1, which we record at the surface. So there are three different components, but you get only one peak. Okay. So coming to N400. So this is a negative potential at 400 milliseconds, and it relates to language. So the N400 is part of the normal brain response to words and other meaningful stimuli, including visual and auditory words, sign language signs, etc. So a large negativity is elicited by sentences which have an anomalous, unexpected, or incongruous ending. For example, Nina takes her coffee with cream, and you expect milk or sugar, but you get socks, so you have a N400 potential over here. So it's negative up. Okay, this one. So it peaks around 400 milliseconds after the stimulus, and the negativity is in the time window of 250 to 500. So what does it mean? It's related to semantic processing and is not just a response to unexpected words. So for this was the original N400 finding by Marta Kutas and Stephen Hilliard, and they published it in Science in 1980. And you have three sentences. The pizza was too hot to eat. That's fine, and negative up. The pizza was too hot to drink. That's a bit weird. So you have this potential happening over here. And finally, the pizza was too hot to cry, which is completely unexpected. So you have this N400. Okay. So where are the electrodes placed for the N400? So again, centroparietal areas. And the subject is seated in front of a monitor, while sentences with anomalous endings are presented one word at a time, 1 hertz. I take coffee with cream and dog. So dog will give you the N400. And here on the right, you see a subject, she's got an electrode cap with probably 64 electrodes, and the different stimuli which are given are John ate broccoli at dinner, and John ate democracy at dinner. So democracy will give the N400. And here you see the N400 effect over here. And most of the action, when you look at a heat map, it's fronto-central parietal. This whole area is activated. So what's its use or clinical relevance? So one is for linguistic research. You can use this as a probe for semantic processing. And the other more practical thing is would be it's an objective screening tool to test for language ability in late primary or early middle school children. So if they can't distinguish between this incongruous or unexpected ending, then you know they have a problem, and the teacher can give them extra attention or exercises. So thank you.